as a Beckman's call it, an amino chain, designed the synthesis of DNA gyrase inhibitors. So there is an emerging problem. Let's look at this news article. Deadly germs largely ignored by drug firms. The number of companies that are producing antibiotics has been steadily declining. This is a result of scientific challenges and business considerations that make finding new antibiotics very challenging. This is a problem. As you see here, the number of drugs that have been approved have been declining over time. And so there's been lots of press releases about trying to fix this antibiotic pipeline because this problem of resistance is on the rise. So you can see here, the number of resistant bacteria is increasing over time, and this is a serious threat in hospitals. So, fact. The structure of gram-negative bacteria makes them much more difficult to kill than their counterpart gram-positive bacteria. This is primarily due to their structure, which has an extra membrane that the gram-positive bacteria doesn't have. And this makes it very difficult for drugs to enter the cell because this extra membrane acts as this barrier. So only drugs with certain properties, physiochemical properties, can actually enter the cell. So in Shaw's laboratory, I'm using techniques in biology and chemistry to synthesize small molecules that can kill this type of bacteria. Fact number 2. Bacteria are very successful in evolving adaptation mechanisms in the presence of antibiotics. They do this through 4 main ways. There's prevention. It's hard for drugs to enter the cell because of this extra membrane. There's modification. You have enzymes in the bacteria that actually modify the drug that prevent it from binding. And then there's mutations of drug targets, so that the target is mutated so that the drug can't actually bind. And then there's efflux. You have enzymes that are actually effluxing the pump and effluxing the drug out of the bacterial cell. So the goal of this project is to find a potent inhibitor that can act as a lead compound for the creation of antibiotics. So this is just an overview of the current classes of antibiotics. There's 3 main classes. You have those that inhibit cell wall synthesis, which is the cell membrane. You have antibiotics that inhibit proteins from being made by inhibiting certain aspects of the ribosomes. And then you have this last class, which is the class that my research focuses on. This antibiotics is focusing on nucleic acid synthesis. So you have antibiotics that interrupt folate synthesis, but also interrupt the enzymes that are involved in replication. And so specifically, the enzyme that I'm focused on is DNA gyrase. And there is a class of antibiotics out there called the quinolones that inhibit DNA gyrase. But the compounds that I'm working on inhibit DNA gyrase in a different location than the quinolones. So I also wanted to show the structures behind the drugs. If you've heard of methicillin, here's the structure behind it, just to give you an idea. And these are the types of compounds that I'm synthesizing. So I'm synthesizing compounds called gyridamides. Gyridamides were discovered in a high-throughput screen to identify compounds that exhibit fermentation in E. coli. This screen was primarily looked for inhibitors of a different enzyme, FTSZ, and it, at the same time yielded these inhibitors of a different enzyme, DNA gyrase. FTSZ is an enzyme that's involved in the late stages of bacterial cell division. And so during this screen, these other compounds were identified because they also exhibited fermentation, which really shows just cell damage or some sort of cell death in some way. So what is DNA gyrase? DNA gyrase is an enzyme that will leave strains as double-stranded DNA is being replicated. So here you see this enzyme, DNA helicase, is replicating this double-stranded DNA, and upstream you have these the strain that's being accumulating upstream so that when helicase reaches that point, they can't continue unwinding the DNA unless the strain is relieved. And that's where DNA gyrase comes in, because DNA gyrase actually cuts 2 strands, relieves the strain, and then renails it back together. And DNA gyrase is known as a type 2 topoisomerase, and that's what they do. And another important implication of DNA gyrase is that it's not present in humans. And you can understand why this would be a very good target for antibiotics, because you'll not target human cells, you'll only be targeting the bacteria. So the background of gyridamide is gyridamide, like I said before, the quinolones also inhibit DNA gyrase, but gyridamides inhibit DNA gyrase at a different binding site than the quinolones. And this is important because they are acting on an unexplored region of DNA gyrase, so there's no confirmed resistance to this mechanism, to that mechanism at this point. And gyridamides, shown here, these are gyridamides produced in previous research, were shown to kill gram-positive bacteria and gram-negative bacteria, but only gram-negative bacteria that are paired with an efflux pump inhibitor. And so the goal of this research is to find a potent inhibitor that can inhibit wild-type gram-negative. So here you can compare the structures of the two types that are inhibiting DNA gyrase. So my project in the lab is to make analogs of gyridamide. Gyridamides are synthesized in 3 to 5 steps. And really, although the structure looks complicated, it's not that complicated. It really can be broken down into 3 parts. Here you have a proline core, and then you have a benzaldehyde and a sulfonyl chloride. And these sulfonyl chlorides and benzaldehyde can be buried at these positions on this core, which is called stable. And that's the structure of gyridamides. So the plan, this is a project I'm working on with a graduate student in this lab. And the plan was to make a virtual library of compounds by varying those 2 positions, varying these benzaldehydes and these sulfonyl chlorides to make a library from commercially started materials. And then apply Moser's rules, which I'll go over in a minute, to try to find the best aldehydes and the best sulfonyl chlorides that can specifically inhibit gram-negative bacterial wild type. And then synthesize this library in lab and test them on bacteria themselves. So this isn't as easy as it sounds, because there are thousands of arylaldehydes and thousands of sulfonyl chlorides, thousands of these starting materials, and we need to try to choose the best ones. So the arylaldehydes we downloaded from Aldrich, single Aldrich, which is just a chemical database. We started with 3,824 starting materials of arylaldehydes, and these needed to be narrowed down into a more manageable size. So, for example, they went through a series of screens, removing different functional groups, such as nitrogen, bromo, different groups that could be toxic in humans or are not found very commonly in medicinal chemicals. And then removed by molecular weight. If it was too large, they were removed, followed by duplicates, and of course, always limited by funding, to finally yield 271 starting materials of arylaldehydes. Then we needed to do the same thing with sulfonyl chlorides. And so here's this is basically summarizing what I just showed you. Here's the 271 arylaldehydes that were yielded, and the same thing with sulfonyl chlorides. So now we have these starting materials to combine in a matrix-like format by putting the arylaldehydes and the sulfonyl chlorides on this ring to yield 3,000 library candidates. And that's obviously way too many to make in lab. So these needed to be further pared down to a smaller size. And that's when Moser's rules come in, and that's what I'm going to talk about next. So ultimately those knocked down the library numbers to 2,000, which were further knocked down to 96 with the final library. So what are Moser's rules? Moser and O'Shea were researchers that looked at the gram-negative antibiotics, analyzing the properties of the antibiotics that are specific to killing gram-negative bacteria. So you can see here in this is the comprehensive medicinal chemistry database. You can see that the gram-negative bacteria occupy a very different physiochemical space than other compounds that inhibit different pathogens and different enzymes that you might look to if you were trying to find a weak compound in one of those areas. So we wanted to narrow this library down by specific properties to killing gram-negative. Some of these include molecular weight less than 600, C-log which measures solubility, and polar surface area. So these compounds were pretty polar. And then narrowed down by diversity and high functional group to ultimately yield 96 gyridamides. And so this is the chemistry that's actually done in lab to make these compounds. Goes through reductive aminations, which is putting the benzaldehyde onto this core, and acylations, so that you can yield this 96 total compounds of gyridamides. So currently, these were sent to collaborators. They were purified at Kansas University and then sent back, and the screening is done here at UC-Davis to determine the minimum inhibitory concentration. It's an assay that shows the minimum concentration that kills bacteria after overnight incubation. And then we can determine the potency and modify. Now, this screen was done on both wild type and drug pump knockout E. coli. So this is just an example of results that I did over the summer. This is one of the, I was doing a positive control, I was doing one of the gyridamides that were found in the previous literature. And my results agreed with the, what the paper said is reported at MIC 5. I found a minimum inhibitory concentration of 4 micromolar, which you can see here. This is what a minimum inhibitory concentration acid looks like. Here's 4 micromolar, the bacteria were inhibited, but only the bacteria that with the drug pump knocked out, and wild type, you can see here, still grew. So there's still research that needs to go into finding more of these compounds that can actually kill wild type. So these are some of the techniques I use, extractions to separate aqueous soluble materials from organic soluble material, chromatography, which is using different purification techniques, and NMR to identify structures, and of course the MIC assays that I just described. And so I just want to thank Professor Shaw for letting me do this research in his lab, and Dr. David Anderson, who's the one we do the bias side with. Jared Moore, my graduate student, and for all of his mentoring. And of course, Professor Louie for choosing me for this program. So thank you very much.
All right. Any questions for our first speaker?
You said you generated a virtual library. Did you actually do that?
Yeah, this is all done pretty much by hand through doing these screens that actually yielded 35,000 virtual libraries.
So you do that with OpenEye software?
It was done through Canaxon. Yeah.
Did you send your compounds away for purification? Why was that?
I think because we have collaborators, it's easier. We were doing kind of synthesizing into something that can be done all at once through a quick purification method to send back, as opposed to synthesizing them all in lab by hand.
Could you comment on the next steps, future goals? How are you going to identify success?
Right. So with the minimum inhibitory concentration, like I said, they were doing it on both wild type and drug pump knockout. And when you use drug pump knockout, that means that the drug can't be effluxed from the bacterial cell. And by doing so, you can see the potency of these drugs. And when you find something if we can find a drug that seems very potent, we can actually modify that particular compound, modify the functional groups to try to make it even more potent. Because the inhibitory concentration that I showed you, like in this example, has an MIC of 4 micromolar. But drugs, really, you want to have something more like the millimolar scale. So by using the drug pump knockout, you can actually determine the potency, and then we can try to manipulate that compound to make it even more potent. So those would be the next steps.
Have you had a chance to try out any of these compounds that you've actually seen?
Yeah, actually, well, this is the first one I've been doing some of these, which have not yielded great minimum inhibitory concentrations, but there's still a lot more that needs to be tested.
We have time for one more question. Seeing none, let's thank Molly and bring our next speaker.